Aditya, and in this video we will study about the structure of chromosomes, cell cycle and cell division. Chapter 2, class 10th ICSE board. So first let's talk about the cell division. What is cell division? Cell division is one of the most fundamental characteristics of life. This is a method which enables life to perpetuate generation after generation. This is equally true in the case of simplest organisms like amoeba, as in the highly complex ones such as our cells or giant-sized elephants or the tall coconut tree. The most conspicuous events occurring during the cell divisions are all related to the chromosomes. The duplicated chromosomes get evenly distributed into the daughter cells during mitosis. Thus, all the cell body cells that are the result of a long chain repeated mitotic divisions have the same type of chromosomes and in the same number. This ensures the normal functioning of the cells and through that the life of the organisms. Now, what are chromosomes? When a normal unstained living cell is observed under a light microscope, its nucleus may not appear to contain any particular thing inside. But when the same cell is stained with suitable dyes, several structures become noticeable in the nucleus. The nucleus contains most of the cell's DNA, which is organized into discrete units called chromosomes. Each chromosome contains one long DNA molecule associated with many proteins. This complex of DNA and protein is called chromatin. When a cell is not dividing, the chromatin under an electron microscope appears to be in the form of very long and extremely thin darkly stained fiber. However, as a cell prepares to divide, the chromatin fibers coil and condense further, eventually becoming thick enough to be distinguished as separate chromosomes. These chromosomes then readily pick up certain dyes and get colored, hence the name chromosomes. Chroma, colored soma body. What are chromosomes? Chromosomes are highly coiled and condensed chromatin fibers. Now, second one, discovery of chromosomes. Who discovered chromosome? When chromosomes in animals were first studied in 1882 by the German scientist Walter Fleming. He noticed these in the rapidly dividing cells of the larva of salamander. The microscope he used was of old type and through it he saw minute threads that appeared to be dividing lengthwise. Fleming called the division mitosis. Subsequently, with the help of more powerful microscopes and by using special techniques, the chromosome and their constituents have been studied in great detail. Now, chromatin. The chromatin material that constitutes the fiber is formed of two substances. First, DNA, about 40%, and second, histones, about 60%. Now, molecular structure of DNA. The shape of DNA molecules was first studied by Rosalind Franklin in 1953. However, the structure was finally worked out by Boston and Crick in the same year. They proposed the stranded helical structure of DNA, for which they were awarded the Nobel Prize in 1962. A single DNA molecule is very large and hence it is described as macromolecule. It is composed of two complementary strands wound around each other in the double helix. Each single DNA strand is composed of repeating nucleotides which are made of three components: phosphate, sugar, arranged lengthwise, and a nitrogenous base attached to the sugar inverts. This is a schematic diagram of helical structure of DNA. Now, which stands to join the complementary nitrogenous base from the other strand. Thus, the two strands together make a ladder-like arrangement with the nitrogenous bases, forming the rungs of the ladder. Four types of nitrogenous bases are found in DNA. These are adenine, guanine, cytosine, and thymine. Adenine pairs with thymine with 2 hydrogen bonds, while guanine pairs with cytosine with 3 hydrogen bonds. That is A equal to T and G equal to C. Now, histone proteins. What are histone proteins? Histones are the proteins that help in the coiling and packaging of DNA into structural units called nucleosomes. The DNA strands wind around a core of 8 histone proteins. Each such component is called a nucleosome. An individual nucleosome can be imagined like a football around which a long rope is wound with one or two loops. A single human chromosome may have about 1,000,000 nucleosomes. Each human cell contains approximately 2 meters of DNA if stretched end to end. However, the nucleus of a human cell is only about 6 meters in diameter. Therefore, the DNA needs to be condensed in order to fit inside the nucleus. The entire chromatin fiber is coiled and supercoiled, something like the coils and supercoils we see in the typical telephone cord, to eventually form chromosomes. Now, figure 2.5 given below this is a highly dramatic representation of the sequential condensation of a DNA strand into chromatin fiber and eventually to a chromosome. Now, structure of chromosomes. Each chromosome in its condensed form, as visible during the start of cell division, consists of 2 sister chromatids joined at some point along the length. This point of attachment is called centromere, and it appears as a small constricted region. Chromosome centromere is located at a particular site. The centromere also serves to attach to the spindle fiber during cell division. As the spindle fiber ruptures, the sister chromatids are separated at the centromere, and each is pulled away from other towards the 2 poles of the dividing cell. After the completion of cell division, the chromatids decondense and revert to their very long and fine thread-like chromatin fibers. There would be as many chromatin fibers inside the nucleus as the number of chromosomes that appear during cell division. Now, what are genes? Genes are specific sequence of nucleotides on a chromosome that encode particular proteins which express in the form of some particular feature of the body. They are the units of heredity which are transferred from parents to offsprings and are responsible for some specific characteristics of the offsprings. Now, just progress check, you can do it. Now, need for new cells. New cells need to be produced for first growth, second displacement, third repair, and fourth reproduction. First point, for growth. Most organisms, including plants and animals, begin their life as a single cell. This cell divides repeatedly to form a cluster of cells which start shaping for different functions to form tissues and organs. Thus, cell division is essential for growth. Second point, for displacement. There is always a wear and tear of cells during the normal body functions. For example, 2 million red blood cells in our body are destroyed every second. These are replaced by new cells formed through a division of their parent cells in the bone marrow. In plants, the old and dried leaves fall off and new ones grow out. Third point, for repair. Apart from normal wear and tear of tissues in the body, there may be accidental injuries. One may get cuts in the skin or fractures in the bone. Repair of such injuries is again through cells which divide, cover up the gaps, and join the broken ends. Fourth one, for reproduction. Reproduction also takes place through the activity of the dividing cells. Amoeba or bacteria just divide to produce two similar independent cells by mitosis. In higher forms, as in humans, or in the banyanti, special cells in the reproductive organs undergo a special kind of cell division to produce sperms and eggs. The sperms and eggs receive only half of the number of chromosomes of their parent cells, that is, one chromosome from each pair. This reduction in chromosome number is very significant. Look at the following example in humans. Now, cell cycle. In order for a cell to divide, it must complete several important tasks. The cell must grow, synthesize materials like protein, RNA, etc., and duplicate its DNA before physically splitting into two daughter cells. Cell perform these tasks in an organized, predictable series of events, which is called the cell cycle. The cell cycle is a series of events that takes place in the cell, leading to the duplication of its DNA and the subsequent division of the cell to produce two daughter cells. Now, a cell cycle consists of two phases. First, a non-dividing phase called the interphase, and second, a dividing phase called the M phase, or simply mitosis. What is interphase? The two daughter cells produced from a mother cell are relatively small, with a full-size nucleus but relatively little cytoplasm. These cells are said to be in interphase. During this phase, they prepare for the next cell division and grow to the same size as the mother cell. Since no changing chromosomes is visible externally during the interphase, formerly it was called the resting phase of the cell, but in reality, it is not true, as the cell is quite active during interphase in synthesizing more DNA. The interphase itself has three phases. First, growth phase, second, synthesis phase, third, second growth phase. Now, first growth phase, RNA and proteins are synthesized and the volume of cytoplasm increases. Mitochondria and chloroplast divide. These two organelles have their own DNA. In late G1 phase, all cells must follow one of the two paths. They may either withdraw from the cell cycle and enter the resting phase, or start preparing for the next division by entering the next synthesis phase. Now, second, synthesis phase. More DNA is synthesized, the chromosomes are duplicated. Third one, second growth phase. This is a shorter growth phase in which the RNA and proteins necessary for the cell division continue to be synthesized. Now the cell is ready to undergo cell division and enter the dividing phase of the cell cycle. After the dividing phase gets completed, the newly formed daughter cells enter into interphase again, and in this way the cell cycle goes on and on. Now, formation of the new DNA. During the S phase of the interphase, each DNA molecule duplicates in readiness for their equitable distribution in the two daughter cells during mitosis. For replication, the DNA double helix open at one end, making the two strands free, to which new strands begin to form, and the process continues in a sequence for the whole length of the DNA. The entire replication is a very complicated process which you will study in detail in higher classes. Can the cell cycle go on endlessly? No. At some places it stops permanently, at some places temporarily, and at other till it is needed. There is a regulatory mechanism for cell cycles. Brain and other nerve cells, once formed in the embryo, do not divide further. Once dead, they are not replaced. Liver cells may divide once every 1 to 2 years to replace damaged or destroyed cells. Surface skin cells are continuously lost and replaced by the underlying cells. A large portion of household dust containing the human skin cells. Now you can study this by your own. You can take the screenshot of this. And guys, if you want the explanation in Hindi, you can tell me. And subscribe my channel also. You can take the screenshot of this. Now the chapter is over. You can read resume of the chapter. Now these are the question answers. Review questions, multiple choice, and you can do it by your own. So guys, thanks for watching, and please subscribe my channel and hit the bell icon so that you will never miss any video. Bye bye guys, take care, stay home.